So when we look at the actual DNA molecule, we're looking at the two posts or the two kind of ends, the structural supports of the molecule. We've got that phosphate bound to the sugar, bound to a phosphate, bound to a sugar, all the way down. And the bond that holds the phosphate to the sugar is a very strong chemical bond. Okay, it is called a phosphodiester bond. Okay, that's spelt like this. And it's very, very strong. All right, so when we look at this chain of phosphates and sugars, they're very structurally solid. It's the difference between making a wall out of brick and making a wall out of sticks. The sticks, that whole idea of the three little pigs, okay, that first house is made of straw or hay or whatever, and that wolf just blows it away because it's not structurally strong. And that last little pig, he makes his house full of bricks. And the poor wolf tries to blow and blow and blow, and it doesn't go anywhere because it's a stronger material. Well, the phosphodiester bond is a very strong bond, and it holds the backbone or the spine of this molecule together. So you've got a phosphodiester bond one way, and then on the other side, it's actually upside down, and it goes up. So it looks like this. So now we've got these two backbones, okay, the two sides to our ladder, really structurally sound. And what's really, really cool about the DNA is what's in the middle, what makes the steps of the DNA. That is what codes for our different genes. Okay, that is what allows for all variety to be there on the planet because of different genetic code, genetic diversity. And it's these molecules that bond in the middle here, okay, that make up those four molecules that I talked to you about, which we're going to learn in a minute. So here's our backbone of phosphate and sugar, and coming off each of the sugars is going to be one of those four nucleic acids. It's either going to be a double-ringed, and I'm going to use different colors here. It's either going to be a double-ringed, okay, there's one. We'll call this one guanine, the G. Then we're going to have another double-ringed A, and we're going to have a single ring T and a single ring C. So if we look at our four nucleic acids, every time we've got a phosphate and a sugar in our actual DNA backbone, coming off of the sugar is going to be one of those nucleotides. So if on this side of the ladder, which one do we want to put coming out of the first one here? We want to put an A? Okay, we can do that. We can put an A there. All right, A. Then which one do we want coming out of this guy here? We want a T? Okay, we'll put T. And then which one do we want coming out of the bottom one? A G? All right, let's put in a G. So here we go. Now, I didn't draw out the structure, but we're assuming that I've drawn out the little molecular structures that go with them. Now, this, again, this same bond here is the same kind of bond here. It's a phosphodiester bond. Very solid stuff. Okay? Very strong. Does that make sense? Now, here's the interesting thing. When we actually look at DNA, because we're constantly making new strands of DNA. We're constantly copying segments of DNA to make proteins, to do this, that, and the other thing. All right? And the way that our body actually, inside the nucleus, the way we actually make more DNA is we consider, all right, we actually consider this to be one package. Okay, this is like one chunk of Lego blocks. And then this guy right here is another chunk of Lego blocks. And this guy right here is another chunk of Lego blocks. And these three molecules together, okay, the phosphate, the sugar, and the specific nucleic acid, these combine to actually be called one nucleotide. All right? And when we actually make DNA, we're blocking together, we're attaching one nucleotide after another. A nucleotide has all three of these molecules in it. It's got a phosphate, a sugar, and a nucleic acid. If we remember that DNA is a ladder, if we look at our image right now, this is only 1/2 of the ladder. We're missing the other side. And depending on what is on this side, that's what's going to determine which nucleic acids and which nucleotides tack on to the other side. So here's our phosphate sugar on the other side. And I mean, sure, I haven't drawn this to scale, whatever, that's fine. For the purpose of explaining to you DNA, I'll forgo that. So if I have a guanine on one side, what has to be on the other side? A cytosine has to be on the other side. G always bonds, okay? G always bonds with C, and A always bonds with T, okay? So if I've got a G here, I know that I have to have a C over here. So this has to be a C here. If I have a T on one side, what has to be on the other side? An A here. If I have an A up here, what has to be on the other side? A T. Okay? Now, we're not done. I'm not done amazing you. Here's the thing. Remember how we said that these bonds are really strong? These phosphodiester bonds? Well, they're really strong because they don't break apart very easily. And they can't. Because remember, these structures are super important. Okay? They're solid. Here's a nucleotide right here. There's another nucleotide. And there's our final nucleotide. Okay? But when we actually go to take DNA apart and to copy different parts, it gets unzipped up the middle. We break the DNA down the middle here, just like a zipper going up and down. And actually, if you can look more closely at an actual zipper, it looks an awful lot like a strand of DNA. Okay, where each little kind of tooth in the zipper meshes in with another one, and you can consider one of these teeth as part of a nucleotide. Okay, now, what ends up happening here between guanines and cytosines, we have these three tiny, and actually I'm going to do them in a different color. We have these three tiny little bonds. And these bonds are very, very weak. Okay, these bonds are called hydrogen bonds. And there are three. So that's hydrogen bonds. A lot easier to break apart than a phosphodiester bond. But between an adenine and a thymine, we only have two little hydrogen bonds. So which one do you think is easier to break apart? Adenine thymine or cytosine guanine? Adenine thymine, because it only has two hydrogen bonds. Okay? So this is an actual strand of DNA. All right? Does that make sense?